Right. I thought to continue from the cytoskeleton. Right. So if you see this cell here, you can see many organelles or subcellular structure or subcellular compartment. I think by now, when you see this drawing, some of the structures you can clearly identify. For example, the middle you can see the nucleus, right? That one is the nucleus, the round shaped one is a nucleus. And then you have learned endoplasmic reticulum. Endoplasmic reticulum is very close to the nucleus. Why is that? Because nucleus has two membranes. Apart from chloroplast, mitochondria, the nucleus. These three organelles, you get the double membrane. So then the nucleus second membrane fold to make the rough endoplasmic reticulum and also smooth endoplasmic reticulum. The difference is rough and smooth is their shape and attachment of ribosome. RER attached with ribosome, SCR no ribosome attached. And RER has flat sheets like this. And then SCR, it's not visible very clear here. It has tube-like structures instead of the sheets. Okay. Now, when we talk about the cytoskeleton, or cyto means cell. Skeleton means, as we know, our skeleton in the body, the bony part, so that give a structural support to our body. Now, we can clearly understand cell membrane is fluidic. It's just like a fluid-like. And inside the cytoplasm, 70% water content. So it's a content of water. So it's like a water ball, just like a water ball. It needs some kind of a supportive structure. So then we don't see a typical picture. We don't see the cytoskeleton because the difference is in our body, the cytoskeleton is made by the bones. Bones are large and very strong, but the cytoskeleton is made by a very thin thread-like structures. We can call microfibers. Micro mean very small fiber-like structures. So we need a very special way to visualize this cytoskeleton. Okay, now you see here, all these pictures, scientists made these pictures with very special dye, fluorescent dye. So fluorescent dyes are a very special color stain dye, and we can use a special microscope to visualize these structures. Now you can see it's like a cotton ball. That means the fibers are large amount of fibers. So they are very thin and they make all everywhere in the cell. So it's a completely packed with these thin the fibers. This is the structure which make the skeleton. So why one function is the cell shape and cell support, structural support. So whatever different kind of cytoskeleton fibers we learn, they have one common function. That is the structural support and the shape of the cell. Otherwise, it's just a fluid ball. So we can give a nice shape to the cell if the cytoskeleton organized like this way, right? Then we have to learn type of cytoskeleton. So there are three types of cytoskeleton fibers. So first you have to learn microtubules. First one is the microtubules. And the second one, microfilaments. Microfilaments. It has another name, actin. microfilaments or actin, that is same. If we see microfilaments or actin, it is the same structure. Then the third one, intermediate filaments. Intermediate filaments. So if you look these three filaments, little bit confusing with these words, because you see a micro, first and second one, it has a micro, and second and third one, it has filament and filament. So sounds like similar, but you have to clearly separate them into three: microtubules, microfilaments, intermediate filaments. Otherwise, when you try to remember these three words, it might be a little bit confusing because of the same words in most of the time. Micro, filaments, filaments. Only the difference is tubulin and the intermediate like words. So try to remember these three words separately: microtubules, microfilaments, intermediate filaments. Right now, first, we're going to see the function of these cytoskeleton. So we can easily remember they have one common function. All three filaments, they have one common function. So what is the one common function? Cell shape. Because we need to remember the function of each cytoskeleton fiber. So some cells are round shape, some cells are tube shape, and some cells are cylindrical shape. You will see so many different shape of the cells, and the cell shape is decided by organization of the cytoskeleton. Right, next, microtubule. The first one is the microtubules. You can see on the screen yellow round-shaped structure. Now you try to assume this round-shaped structure as a 3D structure. That means one yellow color one is just like a ball. One yellow color shape is a one sphere, one ball or 3D shape. And we have a name for this single round-shaped structure. So they are proteins. They are made out of proteins. So this protein is named as a tubulin. That's why the name microtubules, it gives the name microtubules. The tubules is the protein which made this round-shaped structure. Now the round-shaped one unit get together with 13 units. I think one extra here in the picture. So 13 balls joined together and they make a ring. So you can see here ring. And after that, this ring is getting bigger and bigger. It just like here, it's one ring. And then top of that, it's more and more rings adding to that. So one by one, the rings are adding to the structure. Now try to assume the corn, the yellow color corn we boil and eat, maize or corn. So it's something similar to that. If you look this picture, you see, now you can see how we build up the long structure. The 13 rings joined together to make this a long structure. So why it is important? The microtubules, they can elongate, they can extend, they can grow, whatever the distance. Maybe let's assume one side of the cell to the other side of the cell. So one side of the cell is we can call a pole, North Pole and South Pole like the Earth. And then the two sides, the microtubules can make a long line from one end to another end. So they can extend that much by growing these ring-shaped structures. So tubulin is the protein. Tubulin is a one unit. and they join together as a 13 rings around it, and then that is growing up or down, whatever. This direction is growing, and they can make this fiber. So what is in the middle then? So now you look at this picture, the middle is empty. Yes, usually the middle is empty like this. Now this 3D picture, you can see the cross-section 13 subunit, 13 units, 13 tubulin proteins. And then it creates this, the pipe-like structure. So middle is empty, right? Usually in biology, many places, you will learn if something has a empty space, like if it is cylindrical shape, the shape is like a cylinder and the middle is empty. So then the middle area is called lumen. But we don't always use this word lumen for any structures. But here we can use as the middle empty area is a lumen. So it's a hollow shape, like nothing is inside. So microtubules are like that. So we can understand they're like a tubes made out of this tubule, right? Okay, what are the functions of microtubules? So when we're learning functions, it's very important to remember each function of these microtubules. Because in exam, let's say, let's assume like this, there are 3 types of cytoskeletal fibers. There are 3 types of cytoskeletal fibers. Now from each type, let's assume there are 4 functions. One has 4 functions. Example, this microtubule has 4 functions. Then you have to remember the 4 functions. Then again 4 functions, like that. At least 10, 12 functions. Finally, we have to remember for each cytoskeleton fiber. But one thing, they all have a common function. They make the cell shape. They're important to maintain the cell shape. Okay, let's check now one by one the function of microtubules. Now, microtubule has 4 functions. The common function is cell shape for all, right? Then the next thing, cell can move. Not all the cells. Some cells can move. Okay, if you look this cell, that's very special. It has a head, it has a neck, and it has a long tail. So it has a head, and head region, you can see the nucleus. The red color one in the middle is the nucleus. So this is a eukaryotic cell. And then the middle region, then in the middle region, there is a little bit of a cylindrical shape structure, and after that long tail, right? This tail part is known as a flagella. Singular word is a flagellum, plural word is a flagella. Singular flagellum, flagella. So here, actually the right word should be the flagellum, because it's only one tail. So it's called flagellum. Now, what is the function of flagellum? We have to learn coming up lesson. But we want to understand this is helping to move the cell. It's called a movement of the cells. That means the cell move from one place to another place. And then the right word we use for this kind of a thing is cell motility. Cell motility, motile. That's the verb is motile. The cell is motile. Motility is the word given for the movement. Right, now microtubules have to make this flagella. So they can extend from one end to another end, and they make this long flagella. That's one function. Second function of microtubules, cell motility. This cell is known as sperm cell. So we're going to learn in the future sperm cell in reproduction. It's a eukaryotic cell, sperm cell. If you take prokaryotes, like bacteria, they also have this kind of structures because they can also move. Here, this is the shape of bacteria. One type of shape. So they have again these long structures. If you look the first bacterial cell, not this one, the other one, right? This one. So if you see this green color one, they have a small elongations. They're not long, but structurally, the long one and short one, because here you can see the long one here, this one. This is the long one. And the long one is called a flagella. Here, flagella, because it has many flagellums. So it's a flagella. The long one is called flagella. The short one is called cilia. Short one is called cilia. Here the cilia. But structurally, they are same. We're going to learn cilia and flagella again in these lessons. What you need to remember, the cilia is a short extension. The flagellum is long extension. The structurally, cilia and flagella, they have the same, they are same, right? So they are important for the movement of the cells. Right. Next thing is the chromosome movement. Third function is chromosome movement. Now, we learn DNA molecule, but I know that certain theory, 100% it's not going to your mind. When we're learning at first time, it doesn't go into your mind. But when we learn again and again at different places for different purpose, then only you get very clear pictures. If you get a very clear picture, you understand well, it's easy to remember. Because if it's very confusing and you have no idea, you can't remember the structures. Because let's say, example, you're going to learn a language. So you don't know any single word of a language. It's a new language. You don't know any single, but your task is to by heart remember one page. You're going to remember one page by heart, and let's say it's written in English letters. Because let's say German language, 90%, 99%, they use English letters, except few letters are slightly different, one or two letters. They use English letters. Now you have one page to remember, but you don't know any meaning. But you can achieve the goal. If you remember by heart after a few days, after heavy exercise, you can remember it. The second example, you know the meaning of these words. You know the meaning of whole page. You have a clear idea. So that picture, it makes very easy to remember. Probably you spend many days to remember if you don't know it. But if you know it, it's very easy to remember. I think biology, certain areas, they are very confusing. So these confusing areas, we have to slowly analyze and try to understand well. Then only you can remember it. Because remembering is key to get the highest mark. So why I want to tell you that the chromosome. Now, what is the picture you have in chromosome? You can think, what should I know about chromosome? Okay, let's see. First of all, you can see in this picture the DNA molecule. So we learned DNA molecule. It's a one strand joined with another strand, and they join with the hydrogen bonds. After that, they rotate around the axis to make this double helix structure. That's a DNA molecule. Now, how DNA molecule change into chromosome? So why we call chromosome to DNA molecule? So what is different chromosome and DNA? Right, it's like this. The chromosome is a long, I think it's the longest molecule. By far, it is the longest molecule. So this longest molecule made with repeating unit. For example, you know, one strand is made out of nucleotides. So typically in a one cell, one nucleus, there are about 6 billion nucleotide. So 6 billion, one side 3 billion, other side 3 billion. So this one strand has 3 billion nucleotide. Now you can understand, if you take glucose, like glucose molecule can form starch. Glucose forms starch. And starch, again, two types of molecules, amylose and amylopectin. If you look amylose, it is again a linear coil structure, and maybe it is made out of 300, 400 glucose molecule. So we can count in hundreds, we can count in 1,000. Let's say protein is made out of amino acid. So how many amino acid in proteins? Okay, it could be 100, it could be 1,000, but we never talk about millions. So one subunit, one unit is the monomer. This monomer we are counting in 100, if not 1,000. But this is the only place we count in billion, not even million, it's a billion, 10 to the power minus 9. So it's an enormously long molecule. It's very difficult to even think. So if we want to still think, if I take this DNA molecule in a one cell, you know how small is the cell? Like 20 micrometer. How small is the nucleus? Maybe 10 micrometer. So 10 micrometer packed with 2 meter long DNA molecule. The length of the DNA molecule is 2 meters. One cell is 2 meters. Now the 2 meter long molecule should pack into the nucleus. So how we can do that? So we have to compress. For example, from this end to this end, we just want to compress it. So we're going to squeeze this DNA molecule. We call compressed. So when we're compressing the DNA molecule, we can't do only with the DNA molecule. Right? So now you can assume these days you can see kites on the sky, kites in the sky, and they use this ball of thread. If not this cylinder, which is wrapped around the thread. The nylon thread is wrapped around it. So it's a little bit similar. The plastic cylinder is a protein. The thread is the DNA molecule. So a little bit similar way, the proteins wrap around this, the thread is wrapped around the proteins and it compressed. Now this is the chromosome, but chromosome is still not getting the right name. So that means DNA molecule is the first one. Okay, clear, this is the DNA molecule. Second, DNA compressed with the help of protein. DNA compressed with the help of protein. Now, what is the name given to that molecules? That means DNA plus protein. So if we look further, DNA plus protein. Now look at here. This is an electron microscopic picture. In this electron microscopic picture, you can see in the middle a round-shaped nucleus. Ah, yes, this out. Chromatid, yes. The word chromatid, right? Chromatid. Now, okay, I try to say 4 words, right? Listen carefully. DNA, chromosome, chromatin, chromatid, TID, chromatid, TIN, chromatin. Now, 4 words. DNA, chromosome, chromatin, chromatid. Chromatid is TID. Chromatin is TIN. Okay, let's see. Chromatid is okay. Now, if you ask to label what is inside this nucleus, it's not wrong. You can label nucleic acid. It's fine, it's a nucleic acid. So you can also label DNA molecule. It's not a wrong, DNA is not wrong. Nucleic acid is not wrong. But I think the most suitable word is chromatin, TIN, chromatin. Why is that? Because you don't see the structure of chromosome. Because chromosome is X-shape. Usually the picture in our mind is a chromosome is an X-shaped molecule. But you don't see chromosome in this nucleus. It is actually the name given to the situation. Let's say this is the normal cell. And the cell, if we take a picture from the cells using electron microscope, we can see in the middle there are dark areas and there are light areas. There are dark areas, there are light areas. Okay, let's see. The middle there is a round shape, little bit round shape, dark area, right? Maybe this one, I think. It's too. I can't use the cursor. Yes, here, the middle, yeah, this one.
Excuse me, sir, you're muted, sir.
Disconnect, yeah. All right, so thanks. The middle, if you see somewhere a round-shaped structure inside the nucleus, that is a nucleolus. We learn it as a nucleolus. The words are a little bit similar, nucleus, nucleolus. What is nucleolus? It is again arrangement of the DNA molecules, but it has a very special function. We clearly remember the function. Nucleolus responsible to produce ribosomal RNA. rRNA, ribosomal RNA. So ribosome is one of the most important organelle, make the proteins. The nucleus has special place. This DNA give the, we're going to learn that one, nucleus make the codes, the DNA provide the information to make the RNA. That's we keep it, okay, ribosome made by the nucleolus, RNA, ribosomal RNA made by the nucleolus. Okay, apart from that, rest is also filled with the DNA molecule. So these DNA molecules are compressed, packed with the proteins. So we should tell if we want to make an arrow and what is inside, chromatin. Chromatin is DNA plus a protein called histone. Right? It's protein called histone. So we will learn but slowly, right? We can't learn everything at one go, but with the time we have to learn certain thing again and again. We can learn. So chromatin is, remember, chromatin is DNA packed with the proteins. The protein name is histone. Now, what is chromosome? What is chromatid then? Right. You see now, the arrow, check, look at this arrow, the yellow color one. Here. Right. Now, I'm going to show with this small molecule, the record. So I squeeze these DNA molecules to become a short molecule, which you can see pointed with the arrow. That's one DNA molecule. This is one DNA molecule. Now this DNA molecule, at one point, this DNA molecule make a duplicate. Duplicate means a copy of this. So, I mean, the word duplicate is sometimes, it gives the idea that, okay, original one, I have original book. If I make the photocopy of that book, it's like a duplicate. But here we can't use that word duplicate. At the end, both are original. That means DNA molecules make another copy. DNA molecule make another copy. These two molecules are exactly identical. No different. So actually the right word used in science is replication. Because sometimes you see the word replica. There are watches. So replica of that watch. It's not original. It's like a very close to original. We call replica. But the word is called replication. That means replication means one molecules make two molecules. Two molecules are identical. So that's very special for DNA molecule. It's not special for other molecules. Why is that? Because if you assume a starch, it is made out of only glucose. But here different DNA made out of 4 different nucleotides. And they have a special order. The order should be there in the newly made DNA molecule, right? Okay, let's see when it happened, it's like this. Now you see the exactly the copy, the newly made one, copied one. The copied one, now it's called chromosome. So this is called chromosome now. And what is chromatid then? Chromatid is the single molecule is named as chromatid. The two molecules, the two DNA together, it's called chromosome. But the other one is called chromatid. Chromatid is the one DNA molecule in the chromosome. So we can't say which one, both. One, this is the chromatin, the other one is chromatin. And it use sister chromatid. This word is also used sister chromatid. They are same, no different. Now you see DNA, chromatin, chromatid, chromosome, chromatid. So you should clearly remember these molecules and their names based on the situation. Situation means when the cell is rest, cell is working as normal. When we see the picture, it is chromatid. But when cell is dividing, cell going to make another cell, then we can see chromosome. Because that time everything is going to double. DNA molecule double. Then we can see chromosome. Within chromosome, the single molecule is called chromatid. Right? So I think it's very important that you have to remember clearly these words with the meaning, no confusion, because again and again, we are going to learn these molecules. Okay, now a chromosome movement, third function of microtubules, chromosome movement. That means, why this chromosome moving? What is the reason to chromosome move? They are in the nucleus. So it happened during the cell division. That means they separate into two like this. Here, now this is the chromosome, and this one is after separation. And then one is going to one cell, the other one is going to other cell. So here one, it goes to this cell. So this is the cell division. One cell make two cells. And there, the most important thing is the DNA molecules division, DNA duplicating, DNA replication, and separate into the two cells. That means cell division is making two cells. When it makes two cells, the most important event is nucleus divide to make two nuclei. And again, important thing is DNA molecule doubled to give one DNA to this, to DNA to this. We're going to learn cell division, mitosis and meiosis, the two different types in the future. But chromosome is the chromosome should go to the one cell, other chromosome should go to the other cell. So this is possible because these microtubules tie them and pull them to the two ends. So they help to tie them and pull to the two ends. That's a function of, third function of microtubules. Right, the last. Yeah, it's only three, but fourth one is shape of the cell. I think I forget to add one here. Okay. Okay, first one is the shape of the cell. Second one is the movement of the cells. Movement of the cells. Third one is chromosome movement. The fourth one is, yes, yeah, Chitsara, you're right, the organelle movement. Where is that one? I think should be here. Oh, I didn't save that one. Okay, it will come, right? This is something accidentally added. Okay, yeah, here the chromosome movement. Now you can see the chromosome is X-shaped because one DNA molecule copy to make the other one. Just give me a second. I have very important. Okay, here you can see X-shaped chromosome break in the middle, and one is going to the right side, other one is going to the left side. So it just like the microtubules tie them and then pull to this end. So then it is called chromosome movement. So microtubules are important in cell division to do this process, right? The fourth one, the organelle movement. Now, what is organelle? We can collectively say what is inside the cell. So mostly the membrane-bound structures are organelle. That's a general definition. The inside membrane-bound structures are the organelles. But ribosome is not membrane-bound. It is again organelle, right? So it's, the membrane-bound is not very important. The small subcellular structures are organelles. Now, why they should move? Why they should move organelle, right? So what is important of moving the organelle? Are all the organelle moving? Is there any organelle which is not moving? Why it is important of moving? For this question, we can make one good example. Let's say plant. When you take plant cell and we take the leaf, the first layer of the leaf is a single cell layer. That's called epidermis. And below epidermis, these are the RER, yeah, some like a nucleus, RER, they are usually not moving, and even centriole, they just, okay, we haven't learned centriole yet. RER, nucleus, they are not moving actually, but some. If you take this plant cell, the first layer is the epidermis, and under that layer, there's a second cell layer. This cell layer is called palisade cell. And this cell contains large amount of chloroplast. So chloroplast, they are moving within these cells based on the sunlight. If there is a very intense sunlight, the chloroplast can damage. So they have to move out of this intense sunlight. If there's a less sunlight, the chloroplast should move to the top side. So that is an example of organelle movement, right? And a nucleus, you will see, a nucleus is not moving because it is tied to the around it, and then tightly attached to other structures. Nucleus is not moving. Some organelles, they are moving. Even a mitochondria, they can move because they are energy supply unit. So when there is more energy, that area they have to move to provide more energy, ATP molecules. So the four functions of microtubules: cell shape, then the motility of the cell, cell movement, then the chromosome movement, organelle movement. Yeah, cell shape and the strength. Cell shape and the strength. You can take it to the strength and the cell shape. Okay, then second type of cytoskeleton. The second type of cytoskeleton is microfilaments, if not actin. Microfilaments and actin are the same. Let's see how microfilaments made. You can see again yellow color, round shaped structure. This is a protein called actin. This is actin protein. The name of the protein is actin. Microtubules, the protein is tubulin. Microfilaments, the protein is actin. So how it this time actin making these microfilaments? Here they line up like this. It line up, and then again second one also line up. Second fiber is also line up. Filament, first filament, second filament. So two filament then intertwined. So they can intertwine. That means they're going to wrap around each other to make the final version of actin filament, right? The functions, again, cell shape and the strength. That's common to all, right? Second one, muscle contraction. Now you're going to learn muscle contraction in detail, the molecular level, how muscle contract, and there you're going to learn the actin filaments are very important in muscle contraction. At the moment, we are not learning the mechanism of muscle contraction. When you learn in Unit 5 human body, the muscles, skeletal muscles, we're going to learn muscle contraction. So actin play a major role in muscle contraction. Third one, this word, we have a special word, pseudopodia. Formation of pseudopodia. Pseudo is a fake, a podia is an arm. The meaning is a fake arm. So where we can see pseudopodia, why they make pseudopodia, and how they make pseudopodia. Okay, so can you remember lysosome? We learned very special function called phagocytosis. Phago mean eating the cell, right? So phagocytosis, special cells called phagocytic cell. As a special cell called phagocytic cell. This cell is very special. This cell can move their cytoplasm and the cell, but here they don't use cilia or flagella, but they use special movement to move to one side to another side, because all these cells are in blood. We call white blood cells. So these white blood cells are important in immune system, are important in protection against diseases. It's our protective system. Now, if they are in the blood, how can these cells move into the tissues? They should go out of the blood and they should move to the tissues, because the phagocytic cells, they are known as phagocytic cells because of its process. They're going out of the blood and they stay all over our body, protecting our body from pathogen. Pathogen are disease-causing agents like bacteria and virus. For example, if we breathe with the air, pathogen can go to the lungs. Lung is a tissue. So then this phagocytic cell should stay in the lungs to protect the lung tissue from pathogen. So they should go out of the cell. To go out of the cells, they use their whole cell, push the cells, extend one part of the cell, and cytoplasm push into that part. So they can crawl then without flagella or cilia. So they have this ability. This ability is very important. You can see in number 1, pseudopodia surround pathogen. So pseudopodia is the fake arm, that is the extension of the cytoplasm. Cell membrane makes small projections and then push cytoplasm into the cell membrane. I think actin also help to make these projections, because actin filaments, if they can move to these 2 arms, it gives the support. It's just not only the fluid, it has a support. So then they can make this a special trap them. That means the pathogen trap into the cells. And then after that, lysosome, you can see lysosome in second picture, pathogen are engulfed by endocytosis. Now, there are 2 words again, engulfed, endocytosis. So engulfed is trap them. That's not a very scientific word, it's a normal word, engulfed. Pathogen are trapped by endocytosis. So we haven't learned yet movement of molecule across the membrane. So cell has the membrane, the molecule can move out and in with the cells, and that's called a different type of the molecular movement. So this process is called phagocytosis. The cells of the immune system can identify the pathogen. And if you want to find the answer how this cell identify the pathogen, that we're going to learn in immune system. After identify, they take into the cells with the help of lysosome. They digest the pathogen, kill the pathogen, and after that they release the particles out of the cells. To make this, the cellular structures called pseudopodia, actin filaments are very important. And also, if you look this cell, you can see it's not a round. So they have some extensions. They have a cytoplasm make some like a branches like this. So this is a amoeba cell. So amoeba cell can also move with this pseudopodia. So they also use for the movement of the cells. They don't use cilia, they don't use flagella, but they have a different mechanism, the pseudopodium. So they extend the cytoplasm. First they make the elongation of the cell membrane and then push cytoplasm into that. Like a crawling type of movement. So they can still move like that way. So that's a actin role in making a pseudopodia. The last one, 4th one, is a cleavage furrow. What is cleavage furrow? I think the word cleave mean it just cut. The cleave mean just separate. Furrow is some kind of how to say, it's like a groove, like some kind of a projection in. There is a projection in, it's called a furrow. Right. So why cleavage furrow? This word is a very special cleavage furrow, and we can mostly see this cleavage furrow when we are learning cell divisions. Because cell, if you remember cell theory, the 3 principles of cell theory, cell is the smallest and functional living thing. Second one, all organisms are made out of cells. Third one, cell arise from pre-existing cell. I mean, there is a cell like this, and if it divide, we can see two cells. Now, when the nucleus divide inside, in this, let's say this is a cell, and nucleus divide. After that, the cytoplasm make this kind of a constriction, this kind of a movement, and that's called the cleavage furrow. So that's a cleavage furrow. After that, they can separate like this. Okay. So we haven't learned cell division yet. Here, I took this picture from the Campbell. It's a very nice picture. You can see it. First, actin filaments make a ring. Actin filaments make a ring. And the ring is getting smaller and smaller. The ring is getting smaller and smaller. Then this breaks the cytoplasm into two. So here the inward movement is the cleavage furrow, and actin filament make a ring. You can assume like if you know this the one we make called dough. Dough mean when we make in the bread or roti like structures, we make this dough. So if the dough is the soft one, and sometimes you can cut with the thread into two. Something like that, the actin filaments make a ring, and the ring is getting smaller, and it cut from the cytoplasm into two. We will learn this again in cell division. The last one, intermediate filaments. So intermediate filaments, how it made out of, there's no very special, there's no special structures made it, because tubulin make 13 structure and they make the columns. Actin make one filaments and another filaments, and then they intertwine to make the microfilaments. Now, intermediate filaments, many proteins can be made intermediate filaments. One type of protein is the keratin. So keratin proteins can make this kind of a collection. So many keratin proteins make the collection to make the intermediate filaments. Again, cell shapes and the strength to the cell. And second one is anchorage of organelle. Now this time, organelles are not moving. See, to move the organelles, cytoskeleton help. To hold the organelles, cytoskeleton help. One example is nuclear lamina. One example is a nuclear lamina. Nuclear lamina is, you can see the nucleus, round-shaped structure is nucleus. And these intermediate filaments make a strong ring. And this strong ring is tied to the other filaments. Then it can hold in the middle, it's not moving. That's called nuclear lamina. Okay, I think yeah, what kind of a question can be asked? Now, it's very important always. We can remember, we can learn, we can understand, we can memorize. But I strongly believe always we have to get an example. What kind of a question can be asked? Why is that? Because I want that the importance of these things to remember. There are 3 types of cytoskeleton, and they are structured like this, and they have functions are like this. After that, all your knowledge, checking in one MCQ question. At the end, 1% marks coming from that question, right? So you can take time and try to find the answer for this question. Yes, there are two answers. Okay. Now, one of the confusing thing is the wording. I clearly mentioned the name of the cytoskeleton filaments are very confusing because microtubules, first one is a microtubules, second one is a microfilaments or actin. Then the third one, intermediate filaments. This is the place where you make the confusion because not the function of each. The three cytoskeletal words are a little bit similar. So that is an error most of the time. One can make a mistake of marking the right answer. It takes time to familiarize these three words. It takes time to clearly remember these three type of cytoskeleton. Now, first one is a microtubules. The first type of cytoskeleton is a microtubules. And we learn microtubules, they are made out of the protein tubulin. So we have a small helps, this word tubules, tubulin. So we try to remember tubules made tubulins. And then microfilaments are made out of actin. Microtubules made out of tubulin, microfilaments made out of actin. So then the first one is wrong. Microtubules are formed by tubulin. The correct sentence is microtubules are formed by tubulin. And intermediate filaments are made out of keratin. So microtubules, tubulin, microfilaments, actin, intermediate filaments, keratin. So different proteins make these three different structures. So keratin is not present is wrong. Cytoskeleton, keratin is present. Third one, microtubules are involved in movement of organelles. So what are the four functions of microtubules? Shape and the strength, chromosome movement, organelle movement, and the motility of the cell movement. So it's mostly involved in the movements. So microtubules, we learn clearly involved in movement of organelles. So third one is the correct answer. If we try to see what is wrong with fourth and fifth, microfilaments are involved in the movement of chromosome during cell division. No, it's microtubules. Fourth one, microtubules are involved in the movement of chromosome during cell division. Fifth one, intermediate filaments provide a channel to secrete material